This presentation is from Cal Poly Pomona's Bio 2400 Section 1, Group 1, and we will be focusing on a method to minimize ocean acidification by inserting a heat-tolerant gene into mussels.
To 9.6 times. Hello, my name is Hung Vu from Group 1. Here I will explain the goal and rationale of our research. Our presentation is called Reducing Oxide Acidification through addition of heat-tolerant gene in Mytilus galloprovincialis. For many years, our Earth has been facing the phenomenon of global warming. This phenomenon causes climate change, rising the temperatures, and contributes to acidifying oceans. This has affected the habitat of marine animals. In order to adapt and survive in these changing conditions, marine animals have to choose to evolve. For example, the mussel Mytilus galloprovincialis has chosen to thicken itself to withstand the warming of seawater by synthesizing more calcium carbonate from the sea. Here, the issues of interest in our study is that calcium carbonate is seen as a way to combat oxide acidification. The more calcium carbonate in the oxide is absorbed to synthesize the shell of marine shellfish such as mussels, the lower the amount of calcium carbonate used in anti-acidity will decrease, leading to an increase in pH. If we can increase the heat resistance in mussel, the sea will reduce part of the burden of providing calcium carbonate. Therefore, we try to insert a heat stress gene called TAHSP90 found in an Indian wheat into the gene structure of mussel Mytilus galloprovincialis in an attempt to increase their heat resistance. This TAHSP90 gene has been shown to withstand up to 9.6 times more heat and drop than other types and has been applied to develop heat-resistant transgenic crops. We hope that the inclusion of this gene in the mussel genome will partly help to minimize the absorption of calcium carbonate in the oxines, by there contributing to limiting oxide acidification.
My name is Ruby Perez, and looking at the TAHSP90 gene, the diagram shows 3 different restriction enzymes that will be used: XBA1, NCL1, and XHO1. These restriction enzymes will help cut our desired gene into fragments that we can use to insert into our P blue script 2 KS plus plasmid. This gene comes from the species Triticum aestivium, which is a wheat plant, and will help our Mytilus galloprovincialis species of mussels with heat resistance. Once our gene is successfully cut into fragments, it will be introduced into our plasmid P blue script 2 KS plus. This plasmid contains a multiple cloning site, MCS. This area of the plasmid has up to 20 different restriction enzymes. Some of the enzymes included are EcoR1 and BAMH1. Both of these restriction enzymes produce a staggered cut at their restriction sites. These staggered cuts contain sticky ends. Sticky ends make it easier for our gene of interest to be inserted and ligated. The yellow arrow on our plasmid is the ORI site, also known as the origin of replication, and where replication of DNA will occur. The lac promoter is where the RNA polymerase will bind and begin transcription. Our plasmid is also ampicillin resistant. When we begin to replicate our plasmids, those that are resistant to ampicillin will be the transformants, whereas those that don't grow did not successfully implement the TAHSP90 gene into their genome. Taking our recombinant plasmids, they are now ready to be introduced into our Mytilus galloprovincialis zygotes.
My name is Summer Felushkic. I'll be describing the gene strategy for Group 1. We start by finding the coding region of cDNA for the gene TAHSP90 found in Triticum aestivum, cloning it into the SMAI site of a P blue script to KS+, and after cloning, excise the cDNA from the phage vector. The cDNAs are then introduced into the P blue script to KS plasmid, and through further modifications of its material, a fragment of the constructed plasmid will be isolated by preparative electrophoresis performed on an agarose gel and purified by using the gene clean 2 kits, then redissolved in TE buffer. Elsewhere, zygotes are collected from new glochidia, reproduced from a controlled housing of male and female Mytilus galloprovincialis mussels. DNA is injected into the male pronuclei of zygotes after centrifugation of the zygotes. Injected larvae are then transferred into controlled housing of a host fish, such as skipjack herring, for the recipients to adhere themselves to the gills of. The parasitic recipients are then left to grow to term and expelled from the fish as juvenile mussels. After this expulsion, we perform a tail and or flap biopsy of each of the collected juvenile mussels. The integrity of the transgene is examined by southern blotting analysis of genomic DNA extracted from the samples. The recipients are monitored in the controlled environment while growing into adult mussels, to which upon completion will be further tissue tested via RT-PCR, where total RNA is extracted from the tissue with a trizol according to the manufacturer's instructions. The RNA is reverse transcribed, and conditions for amplification of cDNA include 40 cycles of incubation at 3 separate temperatures with primers specific to the TAHSP90 gene. The amplified products are subject to electrophoresis. Mussels with the transgene are isolated from those without and left to reproduce with another set of wild/genetically unmodified mussels. Transmission of the transgene to the next generation of juvenile mussels is examined with southern blotting analysis as conducted earlier.
My name is Brogan Lee, and I will be explaining the strategy reasoning for Group 1. Only the coding region, the exons, of TAHSP90 will be integrated into the mussel genome, as the plasmid will replicate a dysfunctional protein when all the introns of the eukaryotic heat tolerance gene are incorporated. A naked plasmid vector, blue script IIKS+, will be used opposed to other vectors in efforts to keep a low production cost and leave room for a potentially long sequence, as some viral vectors can only carry a short sequence of the desired genome into the host. This plasmid will be introduced into the zygote, as the modified gene will then pass on to all other cells in the Mytilus galloprovincialis mussel. We will then take tissue samples from this transgenic organism and its offspring throughout different segments of the life cycle. PCR must be used in analysis to amplify the gene of interest. Once accumulated, the restriction enzyme digested genome is to be placed in an electrophoresis gel, where with the presence of current, the size of the segments can be found, indicating whether the gene of interest is present or not in the tissue sample. Levels of isoforms of the heat shock protein 70 HSP70 and SHSP families were higher in response to heat stress in Mytilus galloprovincialis and another species of mussel. Heat shock proteins 70 HSP70s are ubiquitous molecular chaperones in the cells of eukarya and eubacteria, used in growth and development programs under both normal and stress conditions, and are easily found in Triticum aestivum.
My name is Sarai Rivas, and I will be discussing the ethical, legal, and moral implications of our project. Given the recent climate change and increasing seawater temperatures, many shellfish have grown to adapt. The Mytilus gallo-provincialis, or mussel, has adapted by forming a thicker shell by absorbing calcium carbonate, thereby leaving the ocean with less. The ocean needs the calcium carbonate to combat acidification. The ethical concerns regarding the marine life and ocean ecosystem is crucial to the development of this experiment. We want to ensure that the species of mussels are not harmed while implementing the heat-resistant gene, as our purpose is to decrease their consumption of calcium carbonate to help decrease ocean acidification. The Environmental Protection Agency will not likely have an issue with the genetic modification of mussels, given that they openly advocate for lessening ocean acidification. We would need to obtain approval by the Food and Drug Administration, who is currently using whole genome sequencing with the purpose of identifying foodborne pathogens. We assume there would not be an issue with obtaining their approval for this project. We would need to prove the effectiveness of our genetic modification through several tests with mussels in a controlled setting to be monitored closely. Overall, we want to cause no damage to the species of mussels or any other species that could potentially negatively impact our goal or our ecosystem.